All right. You guys have any questions over what we were going over last time? All right. We were just starting to talk about DNA. So we were talking about the structure of DNA, how it has this sugar phosphate backbone on each side, and then these nitrogenous bases are the things that actually carry the genetic information. These are called adenine and thymine, and guanine and cytosine. Adenine always pairs with thymine, guanine always pairs with cytosine. And these DNA molecules are really, really long. And so you have to fit all of this genetic information inside the nucleus of a cell, the central portion of the cell where the genetic material is stored. And so it's useful to think about how these things get packaged. So DNA is essentially packaged in chromosomes. So you take a long strand of DNA, and the DNA, when it's in its native form, it's in this helix that you're probably familiar with from high school. And so there's this coil, this twisted molecule that is the DNA. The sugar phosphate backbone is the green part here, and the nitrogenous bases are represented by like the rungs of this ladder. Well, that molecule then gets wrapped around some protein molecules, and then those protein molecules clump together to form the chromosome. And so when you look at a chromosome, this is the chromosome, but it's actually made up of a large number of these histone molecules that have DNA wrapped around them. And the histone molecules are little spherical molecules. They look kind of like this. The DNA is wrapped around those, and then those histone molecules stick to one another to form these filaments, and then those filaments are wrapped around one another again. So we all have cell phones now, but did any of you guys grow up with a phone that hung up on your kitchen wall, had the long curled cord? And when you would answer the phone, you would walk around the kitchen with the phone to your ear. What happens to that cord over time? Well, it starts to stretch out in some cases, but when you hang the phone back up, what does it do sometimes? It gets all tangled at the bottom, and it's because as you've been walking around the room, you've been inadvertently twisting that. So the phone cord has an initial twist, but then as you're walking around the room, you're also twisting it as you talk and walk around. And then when you hang it back up, whatever tension you had on that thing when it was all stretched out while you were walking around the kitchen, you go to hang that up, and that tension is removed. And because you've twisted it a little bit more, this thing then comes back and it twists around on itself, and it creates this impossible tangle. Well, that's kind of what the DNA molecules bonded to the histones are doing. You have this long linear molecule that wraps around the histone, then the histones clump together to form this filament. That filament has a three-dimensional structure that then twists back on itself. And the result of that is that you can pack this really long DNA molecule into a really tight space by basically organizing it this way. And these chromosomes get so clumped up that you can actually see them under a microscope. If a cell is going through cell division, these things even get clumpier and thicker and shorter during cell division. And so they actually end up looking like this. When you go through cell division, one of the first things that the cell has to do when it's getting ready to divide is it has to make extra copies of the DNA, because you're going to take that DNA and you're going to put it into two new cells. So you have to have one copy to go to one cell and another copy to go to another cell. This process is called DNA replication. And you're going to see me represent chromosomes two ways. One way is going to look like this. And when I draw them on the board, I will draw them like two chromosomes that are bonded together at this thing called the centromere. But the way we refer to chromosomes is very confusing. When I draw a chromosome this way, this is basically a chromosome, but it is made up of two chromatids. And just so you know, biology majors find this terminology confusing. So if you're a little confused here at the beginning, don't worry about it. What has happened here is that this chromosome started out as a single chromatid. And when the cell is getting ready to go through cell division, it needs an extra one of these. And so it uses this chromosome to duplicate itself to create another chromosome that looks exactly like it. That's held together with the first chromosome at this thing called the centromere. So in some cases, we would refer to a chromosome that is all by itself, and after the cell has duplicated the chromosome, we have a chromosome that is made up of two sister chromatids that are bonded together by the centromere. During the process of cell division, those two chromatids will become separated, and when they get into the new cells, we stop calling them chromatids any longer, and they are back to being called chromosomes. But this is just something that the cell has to do to make copies of the DNA so that it can partition those copies into new cells. So this is how we would view a chromosome after replication has occurred. So I showed you this figure last time. This is the karyotype. This is all of the chromosomes in the human genome. We have chromosome 1, chromosome 2, chromosome 3, chromosome 4, 5, all the way down to the 23rd set. You can see these areas that don't pick up the dye. This is where the centromere is located. In some chromosomes, it's located in the middle of the chromosome. In other chromosomes, it's located towards the tip of the chromosome. Are these chromosomes that have been duplicated in preparation for cell division, or are these chromosomes that have not undergone duplication? Here's essentially that view, just shown differently. This view here can also be done with what they call GEMSA staining, which then shows the banding, showing you different positions on the chromosome. So this is one view of the genome of humans. This is another view of the genome of humans. Which of these is after DNA replication has occurred? The bottom one, yep. Because now chromosome 1 is represented by two sister chromatids. This chromosome 1 is represented by two sister chromatids. So in this case, we have 46 copies of DNA. Here, we have 92 copies of DNA. But these 92 copies of DNA are organized into 46 chromosomes, each chromosome having two copies of the DNA of that chromosome. Okay? So they actually physically look different because you've gone through this process of duplicating chromosome 1A, duplicating chromosome 1B, duplicating chromosome 2A, 2B. And so you'll see chromosomes in both of these forms, and it's important for you to understand mitosis and meiosis, that the cell goes through this process of building an extra copy and transforming a chromosome from looking like this to looking like this. This chromosome has a strand of DNA on it. This chromosome has a strand of DNA on it, and this chromosome has a strand of DNA on it. But these two strands of DNA should be identical because one was built from the other. So, for example, if you have the gene for eye color is right here on this chromosome, the gene for eye color will be right there on that other chromosome. Now, this chromosome you got from your mom, and this chromosome you got for your dad. Your mom and dad may have different eye color. One of them may have brown eyes, one of them may have green eyes or blue eyes. But if the gene for eye color occurs right here at this dark spot, the gene for eye color for your mom, which is green, will be there, and the gene for eye color for your dad, which is brown, will be there. And so these genes may have different forms, and that's what leads to you not looking exactly like your parents do. These different forms are called alleles. This is actually a term you need to know. So the gene for eye color can have a variety of different forms, but they'll all be found at the same spot on the chromosome of these homologous pairs of chromosomes. So this is homologous pair 1, homologous pair 2, homologous pair 3, and at each point along the length of this chromosome, you will find the same genes on both of those chromosomes. They might not be the same forms of those genes, but they'll be the genes for the same traits. Who has hitchhiker's thumb? Hold up your thumb. If your thumb bends back like yours does, you have what's called hitchhiker's thumb. That's a genetic trait. There's a single gene that controls whether or not your thumb does that, as it turns out. It's found in a particular place in the genome, and it's always found there. It's not found some other place. I don't know what chromosome it's on, but if it's on, let's say that it's on chromosome 14 in this dark band here, you may possess hitchhiker's thumb gene from your mom and not hitchhiker's thumb gene from your dad. That will ultimately determine what your phenotype is, what you end up expressing. But both of those genes will be at that spot in the genome. So this is just a figure that's showing you this. This chromosome 1 has a bunch of base pairs, nitrogenous base pairs. Chromosome 1 gets duplicated, and then there's a homologous pair of chromosomes. One chromosome 1 came from one parent, the other chromosome 1 came from the other parent. And when you stain them this way, you can actually see that the banding patterns on the two chromosomes are exactly the same, basically showing you that the genes that are there are going to be at the same position on all of these, even though the actual trait that is coded for here and here may be different forms. So how does this replication occur as a cell is getting ready to do this? Well, remember that I told you last time that these nitrogenous bases are held together by these weak intermolecular bonds. They're called hydrogen bonds. They're weak bonds. They're not strong bonds like covalent bonds are. And because of that, it doesn't take a ton of energy to split those two strands apart. And if you have an enzyme that does that, it's even easier because enzymes reduce the amount of energy that is required to get something done. And I also told you that because of complementary base pairing, adenine always bonds to thymine, cytosine always bonds to guanine. And so once you have those nitrogenous base pairs separated from one another, it provides the opportunity for other nucleotides to come in there and add themselves to the split open DNA molecule. And so what you basically do is you take an old strand of DNA, which is this blue one, split it off from the other old strand of DNA. So you take this two-stranded DNA and you break it into two single strands of DNA, and then you use the exposed nitrogenous bases to add new nucleotides to create a new strand of DNA. And now you have two strands of DNA where you had only one strand of DNA at the beginning. And it's called semi-conservative because each new strand of DNA, half of it is made up of the old DNA and half of it is made up of newly formed DNA. And this is how we go from having one chromosome to two chromosomes in terms of the DNA content of the chromosome. So this happens, we can think of cells having a life cycle, and this is called the cell cycle. And when a new cell is formed as a result of mitosis, mitosis is how you got to be who you are today. So when you were first conceived by your parents had sex. Sorry if this is news to you. Your parents had sex. When your dad got your mom pregnant, you started out as a single cell called a zygote. And that zygote contained all the genes you need to become who you are now. That zygote went through the process of mitosis to form two cells from the one cell. And then those two cells divided into four cells, those four cells divided into 16, 16 divided into 32, 32 to 64, 64 to 128, 128 to 256, 256 to 512, to the trillions of cells that you are now. That process of cell division by which organisms build up their bodies is done by mitosis. And mitosis, in theory, gives you cells at the end of mitosis that are exactly like the cells that you had before in terms of their genetic component. So your skin cells have the same genes that your liver cells have. Your liver cells, in theory, have the same genes as your kidney cells. What makes a liver cell different from a kidney cell and different from a skin cell is which genes are turned on and which genes are turned off. They have all the genes there. It's just in different cell types and different tissues, different genes are turned on and turned off. But they all have that same complement of DNA. And so when you're going through this form of division, you start off as a new cell as a result of mitosis. You grow to get to a kind of an adult cell size, and then you go through this S phase. This S phase, S stands for synthesis. And this is actually when DNA replication is occurring. So all during this first phase, G1, the chromosomes look like this, single copy of DNA. After you go through the S phase, your DNA has replicated and they look like this because the cell is getting ready to go through cell division. And at cell division, it has to split those two copies of DNA apart, put one set into one cell, put another set into the other cell to make two new cells. So that's why this figure looks like this. So, mitosis. The reason I teach you mitosis is because the real thing that I want you to know is meiosis. Mitosis is a single set of cell divisions. It's involved in somatic growth, the growth of the body of organisms. Meiosis occurs in two phases, two sets of cell divisions. The second cell division in meiosis is basically just like mitosis. So you're going to have to learn mitosis anyway because that's what the second phase of meiosis is like. So I like to introduce students to mitosis first because it helps them understand meiosis. So You guys have any questions at this point? You guys seen this before in high school? You guys have to take a biology class in high school? You covered mitosis and meiosis. Is any of this coming back to you? More or less? Not so much? So mitosis proceeds in a set of phases. The first of these phases is prophase. In a regular cell that is just going through the cell cycle, the chromosomes are really long and stringy. You can think of them as spaghetti strands inside the nucleus of the cell, to the point that you can't identify specific chromosomes. As you enter mitosis, as you enter prophase of mitosis, one of the things that starts to happen to the chromosomes is they do what we call condensing. They get shorter, and as a result of getting shorter, they get thicker so that they become visible. So in prophase, you see shorter, thicker chromosomes. But at this point, the chromosomes are actually going to be made up of two chromatids bonded together at the centromere. And so this is what you see in prophase. The nucleus, which is normally bound by a membrane, that membrane breaks down so that these things are no longer separated from the cytoplasm of the cell. They're just kind of free floating out there. And these things called microtubules begin to form and attach to the chromosome. These microtubules in this figure are these strand-like things that are illustrated here. These things attach to the chromosome at the centromere, where the two chromatids are held together, and they actually facilitate the movement of chromosomes around in the cell. So think of them as like ropes that are tethered to the chromosome. As you leave prophase and enter into metaphase, in metaphase, these microtubule tethers have basically forced the chromosomes to become aligned along this thing called the metaphase plate. They become aligned along the middle of the cell. And normally, when we're looking at a cell, it looks like a straight line, but keep in mind that the cell is three-dimensional. If you were to take this cell that has these chromosomes aligned in a straight line, if you were to take this cell that is spherical and turn it towards the board, it would actually look like a bunch of randomly spaced chromosomes. But if you took this and turned it towards you, these chromosomes would slowly end up being in this plate, this three-dimensional plate. Hello? Hello? Hold on, just a minute, hold on. Sorry, I don't normally bring my phone to class, but I actually do have a case of septicemia developing, and I kind of want to not amputate my leg eventually. So this metaphase plate is a three-dimensional thing. It's a flat plate. When you look at the plate this way, it looks like they're just scattered around. and that's a physical attachment. And the centromere actually acts like an enzyme. And the way that the chromosomes travel along the microtubules is that they actually eat up the microtubule. So the microtubule, you can think of it as being made up of a bunch of little segments. It's attached to the chromosome at the centromere, and there is an enzymatic function here. And when these two chromatids split apart, when the centromere breaks, you will see this chromosome moving in this direction. You will see that this chromosome moves in this direction because there's an enzyme here. And what the enzyme does is it basically eats away at the microtubule. It basically breaks it apart by removing each little subunit. And as it does this, the microtubule gets shorter and shorter. The microtubule isn't pulling the chromosome. The chromosome is eating up the microtubule, and as it eats up the microtubule more, it moves towards where there's more microtubule to eat up, essentially. And in this way, during anaphase, you see the chromosomes look like they're being drugged through the cytoplasm by their centromere. The reason we illustrate them as looking like this is because they're attached to the microtubule at the centromere, and the two arms of the chromosome are being drugged through the cytoplasm. This is what's happening in anaphase. The chromatids have split. These chromosomes are going to one side. These chromosomes are going to the other side. So in anaphase, they move to the opposite poles of the cell. And in telophase, the new daughter cells start to form a new nucleus. The chromosomes become less condensed, and they kind of go back to normal, and you end up with two new daughter cells. So let's look at chromosome count. Here we have two red chromosomes, two blue chromosomes. Now, each chromosome is made up of two sister chromatids, so we actually have 4 copies of red DNA and 4 copies of blue DNA, but they're organized into 2 red chromosomes and 2 blue chromosomes. At anaphase, we take half of each red chromosome and we split it into 2 new cells. So that in the end, we now have, oh, this is actually a misprint. What's wrong with this figure? Oh yeah, this is from a textbook. These should not be duplicated because they have 2 sister chromatids. They should only have 1 chromatid. Do not believe everything you see in a textbook. Now that you know about mitosis, you know that this is a mistake. You might be asking yourself, well, why did Dr. Klawinski choose to put a figure in a lecture slides that has this mistake in it? Because I want to show you that it's a mistake, and to make sure that you know that it is a mistake, because you know something now about how mitosis works. So this is actually a misprint. Each of these chromosomes should be single chromosomes, not the duplicated chromosomes. But point being, in the new cells that are formed, we have 2 red chromosomes just like in the beginning. We have 2 blue chromosomes just like in the beginning. 2 red, 2 blue. It's just that this is showing too many copies of the DNA. So when you read this, as a matter of fact, I'm trying to think, I don't have it in the notes. You'll see that I usually have notes in the bottom of the slides often that explain things that are in the slides. So there's writing down here. But I should have noted in this that actually has a mistake. And the mistake is obvious if you know about mitosis. So just make a note of that. But these new cells that are formed at the end of mitosis should be genetically identical to the cells that you started mitosis with. And so that's the nature of somatic growth. You produce new cells that are exactly like the old cells that the new cells came from. You can look at something like an onion root, and you can see that you have a bunch of cells that are in interphase in either G1S or G2 phase of the cell cycle, but then you have cells that are going through things like prophase. You can see the DNA thickening. You can see cells that are in metaphase, where you see the chromosomes lined up along the metaphase plate. And then you can see cells that are in anaphase, where you can see the chromatids being pulled apart to the opposite ends of the cell. And then you can see this is a new cell. It's building a new cell wall and cell membrane where you see two new nuclei. The DNA, the chromosomes are still pretty condensed. They haven't stretched out again fully. And so you see these two dense nuclei forming in two new cells. Notice that these two new cells are smaller than these adult cells that have already gone through the process of growth. So this is anaphase here. They're pulled further apart. This is the beginning of anaphase. This is anaphase moving towards telophase. So you can actually see these things if you were to put an onion root under a microscope and actually look at it. You can see things that look like these stylized images of mitosis. All right, questions about mitosis? Things you need to know are what are the phases of mitosis, what's going on with the chromosomes at each of these stages of mitosis. You need to know that mitosis begins with diploid cells, cells with two sets of chromosomes, and they end with diploid cells, cells with two sets of chromosomes. In this particular case, there were four chromosomes, so you have two sets of two different kinds of chromosomes. One set of red chromosomes, one set of blue chromosomes. All right, questions? All right, meiosis is somewhat different. It differs from mitosis in a couple of ways. It starts with a diploid cell and ends with a haploid cell, a cell that only has one set of chromosomes. Mitosis has one set of cell divisions. Meiosis has two cell divisions in it. And so we'll label these prophase 1, metaphase 1, anaphase 1, telophase 1, and then we'll also have prophase, metaphase, anaphase, and telophase 2 as well. One of the ways in which meiosis differs from mitosis actually really has to do with prophase, metaphase, and anaphase 1. That's where the real differences exist. So in prophase 1, the chromosomes are getting shorter and thicker. The nuclear membrane is breaking down. These microtubules are becoming attached to the chromosomes, but what's different is that the chromosomes the chromosomes arrange themselves as homologous pairs. So rather than the chromosomes just being kind of randomly scattered around the cell, the chromosomes become attached such that the long chromosome and the short chromosome, the long chromosomes hang out together and the short chromosomes hang out together. So Chromosome 1A will hook up with chromosome 1B. Chromosome 2A and 2B will hook up. Chromosome 3 and 3B will hook up. In this example, we only have 4 chromosomes, so think of this as chromosome 1A and B are hooking up, chromosome 2A and 2B are hooking up. They actually become physically bonded to one another. And this process is referred to as synapsis. Now, weird stuff happens during synapsis. During synapsis, remember that chromosome 1A is these 2 chromatids bonded together at the centromere. Chromosome 1B, I'm going to draw it in a different color, is also a chromosome that is bonded together with 2 chromatids bonded together at the centromere. These things become physically attached. So there's a protein that forms between them that basically physically attaches them to one another. So they're actually connected physically. And I draw them in different colors because one of these chromosomes in you came from your dad. One of these chromosomes came from your mom, which means that if your mom has green eyes, the gene for green eyes is there for your mom, and your dad has brown eyes, and the gene for that is located there. And so they have the same genes even though they can have different alleles for those genes, different forms of that gene. Well, what is weird is you see that in this figure, they've illustrated these chromosomes crossing each other. And what can happen in synapsis is that these ends of the chromosome can get tangled up when they get bonded together. And when they become tangled up, they can break and become reattached. And when they break and become reattached, what was the end of the blue chromosome is now attached to the brown chromosome, and what was the end of the brown chromosome is now attached to the blue chromosome. This process is called crossing over, because the ends of one chromosome crossed over and became attached to the ends of the other chromosome. Now, the interesting thing about this and why this is important is now your dad's brown-eyed genes are associated with a bunch of genes from your mom. And your mom's blue or green-eyed genes are now associated with a bunch of genes that are your dad's. So we now have gene combinations on this chromosome that didn't exist before crossing over occurred. And this can happen anywhere along the length of this chromosome, and it can happen multiple places. So you can have it happening at one end, and you can also have it happening at the other end. You can, in theory, also see it happening anywhere along the length. It's just kind of random. The further away you are from the centromere, the more likely it is to happen, but it's just kind of random. And it's a function of the fact that these things are physically attached to one another, which allows them to interchange portions of the chromosome. So that when you get to metaphase, you get a metaphase plate, but the metaphase plate is made up of paired homologous chromosomes. And you see that this textbook has actually shown them with the crossing over having occurred. These 2 chromosomes are homologous to one another, but this chromatid is now different from this chromatid, even though they're bonded together at the centromere. This is homologous chromosome 2A and 2B. Once again, this all green chromatid is different from this green and blue chromatid, even though they're bonded together at the centromere. Now, what happens in anaphase 1 is these homologs move to opposite poles. So the centromere doesn't split. The two chromatids on any given chromosome stay together, and what you do is you separate the two halves of the homologous pair to each new cell. So you see that this all blue and the green and blue is moving to one new cell, the mostly green and the mostly green and blue moves to the other side, so that at the end of anaphase 1, you get to the beginning of prophase 2, and you have two new cells, but each new cell only has one representative of each chromosome type. This cell has one version of chromosome 1, this cell has one version of chromosome 2. One version of chromosome 1, one version of chromosome 2. So now these cells are no longer diploid. They don't have two copies of each chromosome. They have only one copy of each chromosome. So meiosis 1 is sometimes referred to as a reduction division, because what it has done is it's cut the total amount of genetic material in half of what it was at the beginning. What you see in metaphase 2 is you see something that looks very much like mitosis, except that it's only being done with half the chromosomes. You have the chromosomes lining up along the metaphase plate, but they're not grouped as homologous pairs because the homologous pairs have been separated from one another. In anaphase, you see the centromere disconnecting, and you take the two chromatids and you pull the two chromatids to opposite cells, so that at the end of telophase 2, you have 4 cells. Each cell has a copy of chromosome 1 and a copy of chromosome 2. So you have 4 haploid cells rather than 2 diploid cells at the end of meiosis. But the other thing that you notice is that because of crossing over, chromosome 1 here is different from chromosome 1 here. And it's different from chromosome 1 here, and it's different from chromosome 1 here. Chromosome 2 here is different from chromosome 2 here, which is different from chromosome 2 here, which is different from chromosome 2 here. This act of crossing over produces cells at the end of meiosis that are all genetically different from one another, rather than being genetically identical to one another. How many of you have siblings? Are any of you identical twins? Okay, so you have brothers, sisters. Your brothers and sisters don't look like you. The reason that they don't look like you is that whenever your parents go to make gametes, they're making gametes through the process of meiosis. And the process of meiosis functions in a way that every gamete is genetically unique, such that the odds of you resembling your sisters and brothers in any way is reduced because the process of crossing over mixes up these genes. Associates genes from your mother with your father's genes, associates genes of your father with your mother's genes through this process of crossing over. And so the odds of you looking a lot like your siblings is really, really small, because the gametes that created you and your siblings are all genetically unique, genetically distinctive. This is how populations maintain genetic diversity, is by having systems of meiosis as the way of forming gametes. And this thing that looks like it might be a mistake, which is this crossing over, this breaking of chromosomes, is probably something that has just become evolutionarily fixed in populations because it leads to greater genetic diversity. And in a changing environment, having more diversity in a population is better than having less diversity in a population. And so this is why you all look different from one another, and it's why you all look different from your siblings. So we started off with a single diploid cell, and we ended up with 4 haploid cells. In mitosis, we started with a single diploid cell, and we ended up with 2 diploid cells. So in the same way that you could make a chart, make a table contrasting C3 and C4 photosynthesis, you could also make a table contrasting the differences between mitosis and meiosis. with headings like the number of cell divisions, the ploidy of the beginning cells, the ploidy and number of the end cells. Does synapses occur? Yes or no? Do homologous pairs pair up or not in the two forms? But probably the easiest way is to draw it. If you can draw out this process, you should be able to recreate this on an exam. There's a mistake in this figure. What is the mistake in this figure? Go through it step by step. Prophase, you have the chromosomes condensing. In prophase 1, you have crossing over occur. Crossing over occurs such that you have these different portions of the chromosomes are now associated with one another that weren't before. You divide these out into the new cells. What do you see here and what do you see here? What's that? Nope, this is metaphase, so they're still supposed to be together before they divide in anaphase 2. What's happened to the crossing over in those chromosomes? Crossing over occurs here. Crossing over is still present here. It's happened, and so it's maintained. These things are still the crossed over chromosomes. What's up with these chromosomes? There's no evidence of crossing over. The artist who drew this made a mistake. And after the artist drew this, they didn't give it to the biologist who was the author of the book and said, hey, are these correct? So these chromosomes don't show any signs of having gone through the process of crossing over. Everything else about this figure is correct, but once again, in that particular panel, the effect of crossing over has been deleted. It's gone back to just the regular chromosomes that are all one color. So got rid of the crossing over. The crossing over reappears here, but it's just gone from this particular one. Once again, why do I include a figure that is incorrect? Because it's useful to you to be able to look at a figure and say, this is correct in these ways and it's incorrect in these other ways. What is the reason that it is incorrect? Well, because crossing over occurs in meiosis. Crossing over occurs in meiosis due to synapsis, the bonding together of these homologous pairs back in prophase 1, and this artist just happened to make a mistake when he was drawing the chromosomes. He or she was drawing the chromosomes. Meiosis is how you make gametes, sperm and egg that go to make a new zygote. Mitosis is how you grow a body from a single cell to trillions of cells in the course of the development of an organism over the lifespan of the organism. So two different types of cell division for two totally different types of function. In order to understand genetics, which is the next thing, you need to understand meiosis. Because the first step in doing a genetics problem is to form gametes. And if you can remember what happens in meiosis, the process of forming those gametes correctly helps you with that process. Okay? Yeah?
Once the zygote is formed, how quickly does it split?
Very quickly. Yeah. Hour or two. And then the next one is an hour after that, and the next one is an hour after that. It becomes a very quick process of getting to a single cell to a lot of cells in a relatively fast time period. If you take a course in embryology, back in the day when I took a course in embryology, you study chick embryos because you can open up an egg and you can follow a chick and from zygote to full development. And they time them out in hours. And so we had like 48, 72, 96 hour chicks. And it's striking how much change they go through in the bigger the organism, the more cell division you need to have in order to get the body built up. And so, for example, a chick hatches out in like a week and a half, 2 weeks, typically depending on temperature. Human takes 9 months of gestation. An elephant takes 18 months of gestation. So it just depends on kind of what kind of organism you are as to how long it takes you to get to the point where you're ready to be hatched or born. Other questions? Okay. Hopefully this is review from some class you've gotten back in high school. This is also something that you should have gotten back in high school if you took a biology class, but we're going to review it anyway. So once you have these new cells, you have a zygote and it's going through development, your cells differentiate. All of your cells don't end up looking exactly the same. Your muscle cells look different from your nerve cells. Your nerve cells look different from your skin cells. Your skin cells are different from your liver cells. They're different from your kidney cells. They're different from the cells in your pancreas. So, as I mentioned before, the way in which you get this differentiation among different cell types is not because each cell has a different complement of DNA. It has more to do with which genes get turned on and which genes stay turned off in these different cell types. And so the way this acts is because DNA is used to make a molecule called messenger RNA. And the messenger RNA is the template that is used to build a protein. And in a given DNA strand, there are thousands or tens of thousands of genes on that DNA strand, and there are mechanisms by which a gene gets activated, gets what we call upregulated, gets turned on, essentially. And when that gene gets turned on, the thing that it does is it creates a strand of messenger RNA. That messenger RNA then gets transported out of the nucleus into the cytoplasm. And when it gets into the cytoplasm, the cell takes the messenger RNA and builds a protein based on the code that is in the messenger RNA. So DNA gets turned into messenger RNA. This occurs in the nucleus. Then the messenger RNA is transported out to the cytoplasm, which is where protein is made. And so in the same way that you can duplicate DNA by unzipping the DNA molecule and building a new molecule by putting nucleotides onto the old molecule, you can also unzip the DNA molecule, but you put a different type of nucleotide on that when you're making messenger RNA. DNA, you might recall, stands for deoxyribonucleic acid. RNA is ribonucleic acid. They differ in whether or not they have an oxygen or not. It's still a 5-carbon sugar. It's still a sugar phosphate backbone, but this one is lacking an oxygen that this one has. DNA ends up being a double-stranded molecule, and RNA is generally regarded as a single-stranded molecule. So it generally doesn't complementary base pair with other nucleotides, although this is more true for messenger RNA than another type of RNA called transfer RNA. So this M in mRNA, M stands for messenger, and it's called messenger RNA because it's a messenger that carries the genetic information that is in the nucleus out of the nucleus into the cytoplasm so that a protein can be made using that genetic information. Other ways in which DNA differs from RNA is adenine, thymine, guanine, and cytosine are the nitrogenous bases that are found in DNA. Adenine, uracil, guanine, and cytosine are the nitrogenous bases in RNA. So these are the fundamental differences between DNA and messenger RNA. So what happens is the DNA splits. Once it's split, these nitrogenous bases are exposed. When those nitrogenous bases are exposed, you can add new nucleotides using complementary base pairing. In this case, instead of thymine and adenine binding to one another, which is the case, when you have an adenine, the RNA nucleotide that gets added to adenine is uracil. So cytosine and guanine still bond together, adenine and thymine bond together when thymine is in the DNA, but when adenine is in the DNA, uracil gets added to it. Then, after the new RNA strand is formed, it becomes separated and it gets transported out into the cytoplasm. So the act of making RNA isn't really that different from the act of making new DNA. Same kind of molecular machinery going on, just using different nucleotides. Nucleotides with ribose as the sugar versus deoxyribose as the sugar. Once the messenger RNA is out in the cytoplasm, there's a thing called a ribosome. It's a large protein complex, but this is the thing that makes proteins. Proteins are simply long chains of amino acids. So if you're drinking a protein shake, you're basically drinking a bunch of amino acids. And there are a bunch of different kinds of amino acids. And the sequence of nucleotides in messenger RNA determines the sequence of amino acids in a protein molecule. And so the DNA dictates the sequence of nucleotides, nitrogenous bases in messenger RNA, and messenger RNA dictates the order of amino acids in a protein. And the reason that this happens is that the messenger RNA has nucleotides in sequence. And then there's this other molecule called transfer RNA. Transfer RNA is a type of RNA that does do complementary base pairing. And it does complementary base pairing in a way that there are some nucleotides that hang off the end here on this free end of the RNA molecule. And then we have complementary base pairing that forms this cross-like structure. But then in each of these loops, there isn't any complementary base pairing. And so you have exposed nucleotides hanging off here. And whenever you have an exposed nucleotide hanging off, that exposed nucleotide can complementary base pair with other nitrogenous bases because they're not paired with any other nitrogenous bases. And in this one down here, there's a sequence of 3 nitrogenous bases. This is a transfer RNA molecule. And at one end is the sequence of 3 nucleotides, and at the other end, bonded to these free nucleotides, is a protein, or is an amino acid actually, a single amino acid. And so what happens when this is going on is the ribosome is in 2 parts. There's a big part and a little part, and they clamp around the messenger RNA molecule. So they trap the messenger RNA molecule between these 2 parts. And in this big part of the ribosome up here, there are 2 places where a transfer RNA molecule can fit physically. These are called active sites. And what happens is there are a bunch of transfer RNA molecules just floating around in the cytoplasm, For each of these transfer RNA molecules, there's a sequence of nitrogenous bases at one end, and there's a specific amino acid at the other end. And if the sequence here is guanine, adenine, cytosine, this transfer RNA molecule always carries leucine, the amino acid leucine. If you have uracil, adenine, cytosine, that always carries methionine, another amino acid. So the transfer RNA molecules have this thing called the anticodon, which is this sequence of nitrogenous bases that match up with a particular type of amino acid. And in the messenger RNA is arranged in these 3 nitrogenous base sequences called codons. And the codon matches up with the anticodon, which then determines which amino acid gets added at that point. So adenine, uracil, guanine matches up to uracil, adenine, cytosine through complementary base pairing, and that molecule carries a methionine amino acid with it. This next sequence is cytosine, uracil, guanine. It matches up with guanine, adenine, cytosine. That carries a leucine. This transfer RNA will come in and get in that second spot. Once those 2 transfer RNA molecules are next to one another, the ribosome forms a bond between methionine and leucine. They become bonded to one another. When they become bonded to one another, the bond between methionine and the transfer RNA molecule is broken, and the ribosome takes that transfer RNA molecule out. It goes back out into the cytoplasm, and when it encounters another methionine molecule, it will latch onto that, and it can be reused in the process of building a protein. Once that transfer RNA molecule has been kicked out, there's a space that is left open here. The ribosome moves down a spot. This transfer RNA molecule that was here is now in the first position, and then a new molecule can come into the second position. Methionine and leucine will get bonded to whatever amino acid is formed there. When that happens, the bond between leucine and this transfer RNA will break. This will be kicked out. It'll go off and find another leucine, and then this one will move into the first position. Just does this over and over and over again until you get to a sequence in the messenger RNA that basically tells the cell to stop making protein. And now we're 5 minutes over, and I need to stop. So that's where we're in today. We'll pick this up on Friday.